Hi, everyone. We'll get started in just one moment. We'll wait till to let everyone come in, and then we'll get started. Okay. Hello, and welcome everyone to our weekly journal club. My name is Alice Dalmatakis, and I'm the Senior Lead Scientist for Application Science here at Inscopix. We're excited to have you join us from your virtual offices at home, as it's especially important to stay scientifically connected during this period of social distancing. You can send us questions via the chat window throughout the presentation and for up to 5 minutes after the presentation is over. We will do our best to get to most or all of the questions with a hard stop right at 12 o'clock Pacific time. Today, our translational research scientist, Oliver Miller, will be presenting a molecular psychiatry publication from Wolf Habenstock's lab at the Vienna Biocenter. Oliver did his PhD at Tulane University and Roche Pharmaceuticals in Basel, Switzerland, where he studied the antidepressant effects of ketamine, followed by a postdoc in Zach Knight's lab at UCSF HHMI, where he looked at hypothalamic homeostatic feeding mechanisms using a range of imaging and genetic techniques, including Inscopix miniscopes. And with that, I'll hand it over to Oliver.
Thank you, Alice. Uh-oh, do we have video? Okay, good. All good on the audio? Okay, hey, thanks everyone for being here today. I'm gonna go through this paper from the, oh, I'm getting messages. All good on my end? Okay. I'll be going through this paper that looks at the PVT to central amygdala circuit and its role in regulating passive coping. So I'm new to the PVT, I'm new to the amygdala, and I'm new to passive coping. So this has been a really interesting paper to go through, and I learned a lot. And I look forward to any questions and hopefully some comments at the end to help us all understand this a bit better. So when presented with a threat, animals integrate innate instinctual behaviors with past experiences and adapt behavioral responses to future environmental challenges. The type of responding depends on various factors, but can be classified broadly as either active or passive. For example, when presented with a looming stimulus, mice can choose to respond in one of two ways, either with a rapid increase in locomotion to escape the threat, or with a passive response of freezing. In the absence of any predictive cues, such as the looming stimulus, exposure to an unpredictable stressor, such as escape shock, evokes instead of a cue-directed fear, generalized anxiety state. So a hallmark of such experience is that the organism adapts its behavioral coping strategy away from active escape and towards these passive freezing behaviors. To demonstrate this, the authors implemented a simple assay to test the effects of inescapable stressor on passive coping. So on day 0, mice were tested in the elevated plus maze to establish a baseline of active behaviors and freezing. On day 7, mice were subjected to a 10-minute period of unpredictable, inescapable foot shocks. As you can see on the left panel here, mice who received the shock in red have a much steeper decrease in their active behaviors over the course of the shock period. And then as you can see on the right, conversely, the number of passive freezing behaviors increases over the shock duration. So this is that transition from active coping to passive coping that we talked about in the last slide. 10 minutes later, the mice were again assessed in the in the elevated plus maze to measure how the shock period resulted in changes to active and passive behaviors. So you can see the quantification of this here. Mice who received no shock are in red, and mice who received the shock, no shock is in black and shock is in red. We can see in the group that received the inescapable, unpredictable stressor that there's a decrease in locomotion and an increase in freezing behavior in the second elevated plus maze compared to the first. So these data demonstrate that shock exposure leads to an anxiety-related shift away from active and towards passive coping behaviors. The authors next sought to identify which brain nuclei might be involved in this stress-induced behavioral switch. So to do so, they quantified FOS expression in naive animals, here in gray, and compared them to mice who had been exposed to the elevated plus maze only, and then mice who had been exposed to the shock experience followed by the elevated plus maze. So as you can see in black, the EPM alone recruited quite a few brain regions, but previous shock experience selectively potentiated activity within the lateral periaqueductal gray and the paraventricular thalamus. So given that there's precedence in the literature for its regulation of fear expression, the authors chose to focus on the PVT going forward. So to assess how the PVT is affected by an inescapable shock, the authors used Envista optogenetics, Envista imaging. So they injected GCamp into the PVT and implanted a grin lens into the PVT. Then they attached the Inscopix Envista camera and recorded single cell dynamics while applying an inescapable shock to the mice. So as you can see here in red, there's a subpopulation of PVT neurons that exhibited dramatically increased activity after the inescapable shock, suggesting that the PVT is a relay point for stress signaling in the brain. So to validate their imaging finding, the authors next ablated the PVT using an NMDA lesion. So you can hear with a new end stain, PBS injected mice had intact neuronal structure in the PVT, whereas mice injected with NMDA had fewer neurons. So they successfully were able to ablate the PVT neurons. So you can see here marked in my green bars here, the difference between the blue and the red represents that the ablation of the PVT prevented the transition to passive behavioral coping following inescapable shock. So these data confirm the PVT as a stress sensor in stress-related behavioral plasticity. Again, so ablating the PVT prevented the shock from driving passive coping behavior. So next, they examined downstream targets from the PVT that could potentially be mediating the stress-induced behavioral effects. So given the known role for the central amygdala in fear expression and the fact that we know it's synaptically connected to the PVT, they sought to determine if optogenetically manipulating the PVT to lateral central amygdala circuit could be sufficient for regulating fear expression. So to test this, they injected either chanorodopsin, marked here in blue, or ARCH-T, an inhibitory opsin marked here in red, into the PVT, and then implanted a optical fiber over the central amygdala in order to stimulate those incoming fibers. So compared to control, activation of PVT axons was sufficient to induce passive fear expression in mice naive to shock. So these are mice that have not undergone the shock experience, but simply stimulating the PVT to central amygdala axons is sufficient to drive this passive coping behavior. Conversely, optogenic inhibition of the PVT to central amygdala circuit was sufficient to partially rescue fear expression in mice who had been subjected to the inescapable shock. So yeah, we can see bidirectional modulation of expression of fear behaviors using optogenetics. So yeah, this confirms that the PVT to central amygdala circuit regulates the expression of passive fear behavior. So the central amygdala consists of several classes of inhibitory neuronal populations marked by the expression of different proteins and peptides. So to determine which cell types in the PVT, to determine which cell types the PVT projects to within the central amygdala, they made use of whole cell patch clamp. They patched onto CRH positive, PKC delta negative neurons and PKC delta positive neurons, and used blue light to cause neurotransmitter release and measured the postsynaptic effects. So you can see in all three of the populations, CRH positive, PKC delta negative, and PKC delta positive neurons, there is synaptic connectivity. However, if you're looking at the percentage of responding neurons across those three cell populations, you can see that PVT preferentially innervates these CRH positive neurons within the central amygdala. So based on that experiment, the authors then asked if CRH positive neurons in the central amygdala were actually involved in the PVT's ability to regulate passive fear expression. So in order to do this, they injected the excitatory dread HM3D into the central amygdala in a CRH-CRE mouse, and then applied CNO. And so consistent with this notion, activation of CRH neurons in the central amygdala was indeed sufficient to induce passive fear behavior in the absence of shock. Whoops. And you can see that quantified as the difference between mice who were injected with just GFP alone. Injection of the CNO agonist was sufficient to drive this passive coping behavior. Thus, the activation of CRH neurons nicely phenocopies what PVT activation did, suggesting that this cell type might play a role in the PVT to central amygdala's control of passive fear expression. Okay, so now they get a little bit more into this CRH pathway. So since inescapable shock strongly activates PVT, which then primarily targets CRH positive neurons in the central amygdala, the authors thought that CRH should be locally released from in the central amygdala in response to shock experience. So they tested this by inserting a microdialysis probe into the central amygdala, applying this unpredictable shock and measuring shock-induced release of CRH. So you can see that compared to baseline during the shock exposure, the concentration of CRH within the central amygdala was increased, and that this came back to baseline post-shock. So indeed, activating this shock behavior causes for the release of CRH in the central amygdala. Okay, so then following up on that logic, they infused either CRH receptor antagonist or CRH itself into the central lateral amygdala, and this is in the absence of any shock up here. And then they did the same post-shock. Okay, so which one is this? Okay, so infusion of the CRH1 receptor antagonist, shown in red here, suppress the switch from active to passive coping behaviors after inescapable shock, while having no effect in the EPM-A, which is the pre-shock EPM. And so again, normally in black here, you see that there's this transition to passive coping and that the CRH1 antagonist inhibits that shift in passive coping. So conversely, local infusion of CRH itself, seen in blue, potentiated the shock-induced switch to passive behaviors compared to control mice. One thing that was interesting here, I thought, is that if PVT activating CRH neurons in the central amygdala caused CRH release, if that CRH release was causal for this transition to passive coping, you would expect this blue symbol here to, like this is the pre-shock EPM, you'd expect that CRH by itself should be sufficient to drive passive coping behavior, but in this case, it's not. So they saw that and they thought that there might be some additional local mechanisms within the central amygdala that could contribute to this transition other than just CRH. So they followed up on those experiments next. So to look at those local mechanisms, they patched onto various amygdala cell types in the central amygdala and washed on CRH. So here they patched onto central lateral amygdala somatostatin neurons, and then these are central medial amygdala neurons that are connected to the BLA. So they tested that by stimulating the BLA and recording postsynaptic events in the neurons they're patching. And then they washed on CRH to see what the effect of CRH would be on spontaneous excitatory postsynaptic currents in these cell types. And they found that both in the SOM neurons and in the BLA connected central medial amygdala neurons, that washing on CRH was sufficient to increase the frequency of spontaneous postsynaptic currents. So I made a little diagram just to make that clear because that took me a while to comprehend. So they washed down CRH, which is symboled with these little stars. They hypothesized that these are acting on presynaptic glutamatergic inputs coming into the amygdala, and that activation of CRH receptors was sufficient to potentiate glutamate release onto these two cell types. Okay, so next they wanted to determine which CRH receptor. Okay, so you can see here in green, these are the proposed glutamatergic inputs coming into the central amygdala. They don't know where these inputs are coming from, so that's the next question they wanted to look into. Okay, so to do that, they injected a retrograde tracer, in this case, cholera-toxin B, into the central amygdala of a CRH receptor labeled mouse line. To make that clear, so this is a CRH receptor, CRH1 receptor GFP mouse. They injected the retrograde tracer into the central amygdala. And in this case, they were able to identify which neurons elsewhere in the brain both express the receptor for CRH and project to the central amygdala. So in this way, they identified the BLA and the PVT, as well as the inferior colliculus as being the only brain regions that both project to the central amygdala and express the receptor for CRH. And so those are signified in yellow here. So having identified these nuclei, they next used a flox CRH receptor line. So that's a line that has the gene for the CRH receptor flanked by LOXP sites, such that if you inject a CRE virus into a region, it'll knock out the CRH receptor within those regions. So they did so in 3 different nuclei of the brain: the PVT, the central amygdala itself, and the BLA. And so we can see here, shown in the screen marker that I put in, that selective deletion of the CRH receptor in the BLA is sufficient to prevent stress-induced passive responding after shock. So yeah, thus the CRH1 receptor positive neurons coming from the BLA are a good candidate for this potential glutamatergic input regulating passive peer coping. Yeah, so this is kind of adding on to my last diagram. So now we know that CRH is activating axons coming from the BLA to potentiate glutamatergic input after PVT stimulation. Okay. Okay, so now they wanted to take this model and test it in vivo. So to do so, they again made use of the Inscopix and Vista to record the activity in response to shock of 3 different cell types within the central amygdala. So in this case, they chose the somatostatin positive neurons. So these are ones that when CRH is applied, become more active. These PKC delta positive neurons, which are another common inhibitory cell type within the central amygdala. And finally, an undefined medial central amygdala neuron. And these medial central amygdala neurons serve commonly as output neurons from the amygdala. Okay, so on the left, you have vehicle-treated animals, and on the right, you have CRH antagonist-treated animals. And you can see, just to clarify what context this is in, this is mice who have undergone the shock and are now placed in the EPM. And they've aligned the activity traces of the neurons to freezing bouts within the EPM. So you can see that in these central medial amygdala neurons in the gray trace here, that preceding and during freezing bouts, these cells are highly activated. Okay, so now we can compare that to mice who have been systemically injected with the CRH receptor antagonist before being put into the EPMB. And you can see that this shock-induced modulation of medial central amygdala neurons was suppressed by systemic administration of the CRH antagonist. Okay, so this is kind of the final model we have here, where unpredictable, inescapable stress activates PVT neurons, leading to activation of CRH neurons within the central amygdala, which leads to CRH release, which diffuses throughout the central amygdala, but activates CRH1 receptors on glutamatergic inputs coming in from the BLA, which results then in the passive coping transition. So a really nice, straightforward paper. And with that, we can open it up to any questions and comments. Like I said, I'm by no means an expert in the amygdala circuit nor in fear behavior, but I'll do my best to clarify anything I can, and I'd love to hear any comments.
Great, thank you, Oliver. That was an excellent walkthrough of the paper. I think the authors actually did a really nice job of investigating sort of input by input the role of these various nodes within the amygdala, the role in passive avoidance behaviors. And I think they did a really nice job showing a convincing evidence for the role of CRH on these behaviors. So yeah, again, we encourage everyone to submit questions using the chat box. So we can go ahead and move forward with the question section. So one question is there anything special about the subpopulation of PVN neurons recruited by shock in figure 1?
PVT, right?
PVN, PVT, yeah. Right. So yeah, I think it's
special.
figure 1 with the calcium imaging data.
Let me pop back to it.
I think the next slide, maybe. Yeah, this one.
Something
yeah,
special.
So there's just like, sorry, go ahead.
Go ahead.
Oh no, I was just gonna say there seems like there's a subpopulation that are pretty strong responders. So I guess the question is what may be unique about these cells?
Yeah, so that's a question that the authors did not explicitly follow, but that's definitely a good point. So there's about 100 individual cells being recorded from these animals, and only 20 of those 100 animals were defined as being strong responders. And I think they said that was a delta Z of at least 2, whereas the majority, the other 80 of those cells were either weak or non-responders. My best guess would be anatomical segregation. PVT is very much a relay hub. It receives inputs from many, many nuclei in the brain and sends outputs to many, many nuclei in the brain. So potentially this is the shock sensitive subpopulation that then projects to central amygdala.
Yeah, I think it'd be really cool if there was a way to, like, after you record from the cells, be able to sort of define them by, like you mentioned, maybe a projection specific manner, and then see if it sort of parses out that way. Because I think, yeah, that's probably the answer, but to be determined.
Definitely. I left out one figure just for brevity, and that's where they injected colotoxin into the central amygdala, put the mice through the unpredictable shock, and then looked at CFOS in PVT to determine if it's in fact the PVT to central amygdala neurons that are being activated, and that was well confirmed. So there was like very nice overlap between the retrograde tracer from the central amygdala and FOS, and there was FOS in other cell types as well, but it seemed like there was like very strong overlap between the central amygdala projectors.
Yeah, so I guess since we're on this slide in particular, one question is, is the PVT input to the central amygdala VGLUT2 positive? Is that known?
I believe that's the case. I could be mistaken, but I think most of PVT is glutamatergic. So I would expect that it's VGLUT positive. They also did that Crackham experiment where they patched cells in central amygdala and and applied blue light to drive synaptic stimulation.
Yeah.
This is a holding potential of -70, so these are putatively glutamatergic, although they didn't do the pharmacology to verify that.
Right. Yeah, I guess maybe a comment came in that these VGLUT2 neurons from the PVT also are CRH positive. So maybe there is a circuit from VGLUT2 positive CRH positive neurons that target CRH positive neurons in the central amygdala. That's possible.
Right. So a secondary source of CRH.
Yeah.
Right, and that's interesting with what we know about how peptides are released. Typical neurotransmitters, a single active potential is sufficient to cause synaptic release, but there's some, I guess, semi-controversial ideas that it requires repetitive stimulation to drive release of neuropeptides, and so potentially there could be some cool interplay between PVT, like frequency of activity, driving glutamate release versus CRH release, and and also that interplay with CRH positive neurons in the amygdala.
Right. Yeah. Okay. Another question: Are the behavioral impacts of the various manipulations restricted to freezing versus escape, or are there more complex effects?
Yeah, I kind of tried to respect their verbiage, but I think a much simpler way to have described this paper is distance traveled versus time freezing, rather than active and passive coping. It would have been more straightforward for me, at least, to just have it defined as such.
Yeah, I guess another question sort of along the same lines. So this paper did a nice job of parsing out the circuitry involved in passive avoidance behaviors. Is there anything known about how these circuits are involved in active avoidance behaviors?
Yeah, again, I'm not a amygdala pro, but there was a bit of discussion about, so yeah, I started talking about looming behavior, and so this is like cue-based, and similarly, like fear conditioning is cue-based, so you you pair a tone with a shock, and you have this predictive cue that signifies incoming stressor. And I think the central amygdala is much better defined in its role of mediating these like more Pavlovian fear responses, these cue associated ones. So I think that's really a big step forward in this paper, is that this is defining a role for the central amygdala in non-Pavlovian behavioral plasticity. It's
actually a great segue to the next question, which is, In figure 1, the authors show an increase in PV activity to foot shocks. Would we expect PVT neurons to also respond to cues that predict foot shocks? Is there any learning involved in this brain region, or do we think it's simply relaying the unconditioned aversive stimuli information to the central amygdala, and then that's actually where the learning is taking place?
That's a really good and interesting question. Yeah, I think there's a lot known about central amygdala's role in this like plastic or in this cue related fear. I don't know about PVT.
Yeah, it would definitely be interesting to look at for sure. I think we're finding out more and more that there's actually very few brain regions that are just relay structures. So something could be going on there as well, upstream of the amygdala.
If you look close enough, you're gonna find some cool plasticity everywhere.
Yeah, exactly. Okay, so let's see, another question. Do you think transfecting arch and V-glute2 positive neurons from PVT could have the same effect as antagonists of CRH on coping behavior? Is that a possibility?
I might have misunderstood this question. Can you say that again? So in this case, they put channelrhodopsin and ARCH-T in PVT.
And PVT, yeah. V-glute2 neurons from PVT. Right, so in this case, they were looking at terminals from PVT to central amygdala. So yeah, I think this is maybe what the question is asking, which is something that they already did, unless I'm misunderstanding the question too. Yeah.
I might have gone through this one too quick, but yeah, the activation of this PVT to central amygdala circuit in the absence of any shock was sufficient to drive passive coping. And then conversely, mice who had experienced the shock but had inhibition of this circuit, the inhibition of the circuit was sufficient to like inhibit the transformation into passive coping.
Okay, so I think they meant inactivating the V-glute2 input could have the same effect to antagonists in presence of foot shock.
So we can compare this figure to that CRH figure. Yeah, exactly. So this is the red, this is the CRH antagonist. The red one to the right here is application of the antagonist with the foot shock. So that inhibited transition into passive coping.
Great. So I had maybe not a question, but a comment. Can you go back to the CFOS figure?
Yeah, where is that? Wrong way. Getting there. No. Where was that?
It should have been figure 1.
Went way past.
Yeah. Oh, next. But yeah, here.
This one. Gotcha. I was looking for fluorescent cells.
So I thought it was actually pretty interesting that the elevated plasmase alone actually caused this pretty robust CFOS expression in all of these different brain regions associated with fear and stress, maybe suggesting that the elevated plasmase apparatus is in and of itself anxiogenic. Is this, I don't know, what are your thoughts on that? Like, is this to be expected?
I guess that's not a huge surprise for me in terms of, you know, mice like to be in dark little corners, and this is like a pretty high lux light environment and has unprotected sides. So like this balance of curiosity and fear, like the mice are still exploring the open arms, but are nervous. And so I'm not surprised that activates like the PBN and other stress related circuits, I guess.
Yeah.
I thought this was a really nice way of getting at their question, actually, is like, let's compare just exposure to the apparatus. So like some sort of enrichment or some sort of stressor, and then apply the shock in another group and really parse out like generalized stress from stress that results in passive coping. I thought this was a cool way to get out.
Exactly. Yeah. So not just using the elevated plasmase to be a measure of like an underlying anxiogenic state, but it, as we were saying, in and of itself is actually anxiogenic. Just the fact that it's, elevated and bright and all the things that mice don't like. Okay, so another question. So this study points to a role of CRH in anxiety. Are there currently any therapeutics out there that target CRH receptors for anxiety disorders?
I do not know. That's a super cool question, and something I'm definitely gonna Google after this is over.
Yeah.
But yeah, I'm not aware of. Well, I think that could also be tricky. Like this is looking at, one circuit in its and how it uses CRH to regulate fear and anxiety, but CRH is a really broadly expressed neuropeptide that acts on
absolutely
pretty much all of the brain, as far as I know. So I'm not sure if that would be a good pharmaceutical target.
Right. Yeah, good point.
But I think it's a cool question.
Okay, so I think those are all the questions that have come through. So I think we'll go ahead and wrap this up here. Thanks again, Oliver, for walking us through this really nice publication. Thanks everyone for attending and sending in questions. I think we had a really great discussion at the end. We hope that this was helpful for everyone and that you'll join us again next week. We will be reviewing another interesting publication at the same time next Friday. So please stay safe and healthy, and hopefully we can all get back to our regular research activities soon. Take care.